Chapter 3, Cell Structure and Function. A cell is the smallest unit of a living thing. They're the individual building blocks of all organisms. They exist in two general categories, prokaryotic and eukaryotic. On the far left, you can see a nasal sinus swab from a human. In the middle are highly structured onion plant cells, and off to the right, you can see some bacteria. We'll examine all three of these before the video is done. Microscopy has been developing over time since the 1600s. They started out as simple ground glasses, and now they exist commonly in science labs and much more technical forms, as seen here. Microscope A is a light microscope. It uses a light source and multiple lenses to make thin slices of dyed material appear from 10 to 1,000 times larger. The scope on the right, scope B, is a dissecting microscope. They typically magnify whole objects about 20 to 80 times larger using stacked lenses and bright light sources. Electron microscopes, which are considerably more expensive than those previously discussed, work in a very different fashion. When using an electron microscope, samples are prepared by a thin covering of metal, usually gold, and a beam of electrons is shot at the sample. The electrons bounce back off the sample and into detectors. That creates a refraction image, like the one seen here. We've used what we learned from years of studying histology in tissues and other cells to screen for illnesses and help mankind in a variety of ways. Pap smears, as seen here, are just one example of this. The outcome of literally hundreds of years of examining cells is the unified cell theory, something that's still supported today. Published in the 1830s, it stated that all living things are composed of one or more cells. Those cells can be considered the basic units of life, and that all cells arise from existing cells. Let's dig into the study of those individual units of life. It turns out they all share four component parts. Having a plasma membrane that surrounds them, protecting the cell's interior, is the first. The second is the existence of a cytoplasm, or an internal jelly-like region, where different organelles are found. They also all have genetic material, and they all have ribosomes, which allow that genetic material to be turned into usable functional proteins. A prokaryotic cell is a simple, single-celled, or unicellular organism that lacks a nucleus or any other membrane-bound organelle. They're incredibly simple when compared to eukaryotes. They have those four components previously mentioned, plus a few potential specializations. They can be wrapped in a cell wall in some instances. They have pili, which help them to move slowly or pass material over or around their bodies. Those that need to move quickly have long whip-like tails called flagellum. Prokaryotes are very, very small. In some instances, an entire prokaryotic cell can be smaller than some of the organelles that exist as just a single part of an eukaryotic cell. This chart allows you to compare the sizes to wrap your heads around just how tiny we're talking. Let's move to the typical eukaryotic animal. Here you see an animal cell with individual labeled organelles. We'll walk through these one by one in upcoming slides. I just wanted you to see an image first. Now let's take a look at a typical plant cell, and then we'll head into that list. Pause the video here and really look. Write down what you note the differences are in their overall structure from the animal cell you just saw. Let's run through the basics of cell components and their functions. The plasma membrane separates the cell from the external environment and controls the passage of all molecules that are coming and going. All cells have a plasma membrane. The cytoplasm provides structure to the cell. It's the site of quite a few reactions, and it's a medium in which the organelles are housed. Think about something sitting in packaging peanuts in a box. The cytoplasm would be the peanuts. The nucleoid region is only found in prokaryotes, and that's simply the location of DNA. It's not wrapped in anything special, but because the DNA consists of a clump, we give it a name. The nucleus is a cellular organelle that houses DNA and protects it in eukaryotic cells. It also is the site of directed synthesis of ribosomes and certain proteins. What's a ribosome, you might ask? A ribosome is actually a specialized piece of RNA that's capable of making proteins. You find ribosomes in all types of cells because technically it's not an organelle because it's not membrane bound. The mitochondria is the organelle that everybody seems to know. It acts in ATP production through a process called cellular respiration. Peroxisomes are organelles that oxidize and break down molecules and turn them into peroxide. Vesicles and vacuoles are organelles that help in storage and transport. Centrosomes are specialized organelles that have an unspecified role in cellular division in animal cells. They organize all the microtubules in the cytoskeleton, but we don't quite understand how they do that yet. Lysosomes are kind of like peroxisomes. They also digest macromolecules and can recycle worn-out organelles. We only find lysosomes in animal cells, just like the centrosomes. Cell walls, however, are only found in bacteria and plants. They act in protection, structural support, and the maintenance of cell shape. Chloroplasts are also only found in plants. They're the sites of photosynthesis. The endoplasmic reticulum, found in both plants and animals, modifies proteins and synthesizes lipids. The Golgi apparatus, found in plant and animal cells, modifies, sorts, tags, packages, and distributes both lipids and proteins. It acts as quality control and shipping for the cell factory. The cytoskeleton, found in all cells, maintains their shape, helps them to keep their internal structures organized, and allows them to move them around. Flagella can be found in all three categories of cells, but they're not super common, so we don't give them solid yeses across the board. Cilia are also not super common, but they do function in cellular locomotion, like moving particles along the extracellular surface of the plasma membrane and helping to filter things. You can find cilia in your lungs and along your alimentary canal. It pushes things out of the body, or maybe you can think about the cilia in your nose that help you to filter out dirt. Now, we blew through those pretty quickly, so let's take a closer look at some of them. This is a slice of plasma membrane, and it's made of a phospholipid bilayer. The glycerol heads with their phosphate groups are hydrophilic and point towards the outside of the cell and the internal cytoplasm. The hydrophobic tails point towards one another and create a barrier that is sandwiched between hydrophilic layers. There are proteins with various functions spanning the membrane. Some stick out towards the middle and some jut out into the extracellular matrix. We'll study these more in just a bit. Cholesterol can also be found in the plasma membranes, and occasionally will even run across glycoproteins and glycolipids. We'll get to their function in just a bit too. Here you can see a drawing of the cytoskeleton. Note the general locations of the different types of filaments. Take a second to sketch this out. Use individual colors if you can. Microfilaments are the smallest and help hold the external shape of the cell. Intermediate filaments anchor things in the internal space, and microtubules are shown here in these odd branching patterns called asters because that's what they look like when the cell divides. Here we meet the nucleus and part of the endomembrane system. We'll work our way out from the middle. The nucleolus is condensed in the center. That's where you'll find a lot of your ribosome-making machinery and the DNA you're currently working with. They label chromatin. Chromatin is just a name for DNA when it's packaged with special proteins called histones. It's nice and stretched out, and you can work with it when it's chromatin. It's not as tightly packaged as chromosomes, which is what most people think of when they first think of DNA. They show you the nucleoplasm, which is kind of like the cytoplasm inside of the nuclear envelope. That nuclear envelope, it controls passages into and out of the nucleus through highly controlled channels called nuclear pores, or nucleosomes. Just like the gatekeeper system with the plasma membrane. Here they show it butted right up against the rough endoplasmic reticulum. It's rough due to the ribosomes that dot it. These connected sacs are the perfect place to ensure proper protein folding and configuration. You could also add in some smooth ER if you wanted to. In the smooth ER, you make lipids, store calcium, and pull apart different toxins in the body. Products from the smooth ER and rough ER head over to the Golgi body. This is a transmission electron micrograph that's up close and personal with the Golgi body. Those semi-flattened rings are its membranous sacs. All of those vesicles have been packaged and shipped from among them. Speaking of vesicles, here's the process phagocytosis, which is essentially cell eating. The bacteria in this case is being destroyed by a macrophage, or a white cell. The bacterium is brought into the cell and wrapped in a membrane to isolate it, and therefore, technically, you could call it a vesicle. It's then paired with a vesicle containing digestive enzymes known as a lysosome. Just like Lysol cleans your kitchen, the lysosome cleans up the bacteria by digesting it out into its component parts. To review the endomembrane system, you have messages coming from the nucleus in the form of RNA that direct protein synthesis in the rough ER. Proteins made in the rough ER can be transported in vesicles to the Golgi apparatus. Vesicles always enter the cis face of the Golgi. After they're worked on, they are transported from the trans face of the Golgi. They can link up with the plasma membrane and have their internal contents dumped to the extracellular surface of the cell. Then your body can transport them wherever they're needed. Here you can see another organelle, the mitochondria. There are two membranes in the mitochondria, both the outer and the inner. The outer comes from the cell, and the inner membrane is actually manufactured by the mitochondria. It carries a little bit of its own DNA. The inner folds, they point out, increase the surface area, and they're called cristae. There's also an inner cytoplasm-like substance called the mitochondrial matrix. It's within the individual mitochondria that glucose molecules are turned into adenosine triphosphate, or ATP. That's the energy molecules that all cells use to do work, and we'll get to know them quite soon. Here's another energy factory found in plants called a chloroplast. It has an inner and outer membrane that we'll get to know quite well, just like the mitochondria. There are internal structures called thylakoids that sit in stacks called granum. The aqueous fluid trapped in the middle of a chloroplast is called the stroma. We'll get to know all of these individual pieces and why they are important in a chapter coming up. The chloroplast's main job is to turn light into sugar, which is a very interesting process to study. The outside of a cell can be just as important as the inside. The extracellular matrix consists of a network of substances all secreted by the cells themselves. There are collagen fibers and strands with carbohydrates, proteins, and polysaccharides. These molecules can help hold cells together, contribute in their communication, and act as a cushioning or padding between the cells themselves, giving a tissue a little bit more bounce and therefore more resilience to external pressures. Holding cells together is an important job too. There are 4 kinds of connections between cells. In A, you see a plasmodesmata, which is a channel between cell walls of 2 different adjacent plant cells. They can share water and other fluids freely. In B, you see the tight junctions that join adjacent animal cells. Tight junctions are important when you don't want fluids moving outside of cells or outside of the boundary that they create, like your stomach acid. You want to keep that trapped. In C, you see desmosomes, which join 2 animal cells together, but not necessarily in this fluid-tight configuration needed in the tight junctions. Desmosomes can help keep your skin cells together or even your muscle cells. Gap junctions in D are special junctions. Gap junctions act as channels between animal cells when quick communication is necessary, but it also might need to be turned off by the closing of that little tiny connexon. You find gap junctions in your heart. Your heart has to be able to share messages to beat very, very quickly, and thus it uses gap junctions. Now that we're familiar with the cell, let's look at a couple things it's physically capable of doing. Here's another close-up of a plasma membrane, but with a focus on different types of various proteins. The main component of the plasma membrane are those phospholipids, but proteins actually make up the second major chemical component. Integral proteins are embedded in the plasma membrane. They can span the whole thing or just part of it. Integral proteins serve as channels or pumps to move materials into and out of the cell when necessary. Peripheral proteins are only found on part of the exterior or part of the interior. They can be attached to other integral proteins, or they might have some phospholipids anchoring them. Both integral and peripheral proteins can serve as enzymes to speed up chemical reactions. They can serve as structural attachments like rivets, for the fibers of the skeletal system, or they can serve as part of a cell's recognition site. Carbohydrates come in third place when it comes to major components of plasma membranes. They're always found on the outside, bound to either proteins or lipids. These carbs can be between 20 and 60 monosaccharide units in length and either be straight lines or highly branched structures. Along with peripheral proteins, carbohydrates form specialized sites in the cell surface that allow cells to recognize one another. When cells come across something they don't recognize, they signal the immune system that there's an invader. This is how we know to attack and digest bacterial cells. If you make mistakes in recognizing invaders, your body can develop an immune system disorder, like lupus. Something important to the health of the cells are the movements of molecules into and out of them. Movements of ions and molecules that aren't water is referred to as diffusion. Diffusion through a permeable or open to movement membrane follows the concentration gradient of a substance. The substance moves from an area of high concentration to one of low concentration. If you've ever put a drop of food coloring in water, you've seen this before. The plasma membrane of a cell is permeable to very small nonpolar molecules like oxygen and carbon dioxide. Your cells don't have to think or plan the movement of these gases that are critical to their function. Osmosis, on the other hand, is the movement of water. In osmosis, water always moves from an area of high water concentration to an area of low water concentration. In this system, the solute cannot pass through a selectively permeable membrane, and because water likes to follow solute, you end up with these odd height differences. So you have the same concentration of water and solute on both sides. The movement of water into and out of cells directly affects their health. Water is drawn to solutes. It will follow it. We have different terms to describe solutions that regularly affect cells. Let's look at how animal cells react to certain solutions. A hypertonic solution is one with a very high concentrate of solutes or ions. As water follows solutes, it will leave a cell that's sitting in a hypertonic solution, and the cell will become wrinkled and shriveled. In a hypotonic solution, the solute level is very low. As there are more solutes in the cells themselves, the water will actually move into the cell towards the solutes, and the cell will fill up like an overfilled balloon, and in some cases it can burst. Isotonic solutions are solutions in which the solute levels match the inside and outside of the cells, so the water level remains even and correct continuously. It's a little bit different in plant cells. In a hypertonic solution, a plant cell will again lose water, but the cell holds its original shape. There's a membrane on the inside though, and it's going to collapse. You just don't quite see the same damage though that you do in animal cells. In an isotonic solution, water will regularly move into and out of a cell, just like it did in an animal cell. However, plants are actually built to prefer the hypotonic solution, the low concentrate. They like to keep a full central vacuole in their centers. This allows them to exhibit turgor pressure, which keeps them tall and healthy and allows them to grow to great heights even though they don't have any firm molecules to allow them to reach those heights. Here's a close-up of the movement of certain solutes and water across the plasma membrane. There are some gradients that are very important to the functioning of cells. Notice that there's a balance of sodium, chlorine, and potassium. That's the K's in this picture. Their charges are marked because they must also be balanced. Remember that opposites attract, so there's a lot to consider in this particular electrochemical balance. The body takes care to keep potassium inside of the cell, creating a very electrically positive space. The outside of the cell has a very high concentration of sodium. Special pumps move potassium and sodium ions across the plasma membrane. Why does this matter? When you have both an electrical and chemical gradient, ions will move incredibly quickly to balance it out. This is called equilibrium, having everything be equal on both sides. These quick changes in the electrochemical balance are the way your nervous system cells relay signals across their membranes at lightning speed. This is why your nerves can speak to each other so quickly. As the final topic of what a plasma membrane can do, it's capable of helping the cell both take in and send out particles that are large, small, liquid, or solid. Here, we see 3 variations of endocytosis, or into the cell. One is phagocytosis, or the bringing in of large particles. Large particles can be wrapped up in the plasma membrane and put into vesicles. That particular vacuole is probably going to be met with a lysosome. In the middle, you see pinocytosis, and you can think of pinocytosis as cell drinking. The cell membrane simply creates a little pocket in which it can take fluid. Once it's filled up with fluids, it pinches off and brings that fluid inside of the cell. In C, you see receptor-mediated endocytosis, and I've always thought of this as cell sipping. You have little tiny Catherine-coated pits, and they're looking for specific, very small substances or molecules that are in the extracellular, or that are in the fluid around the extracellular membrane. Once those tiny receptors get their hands on whatever molecule they'd like, they signal to the cell to bring that Catherine-coated pit inside of the cell with a nice high concentration of whatever molecule they were looking at. Your cell can be very specific about what it brings into itself. It can also be specific about who it kicks out. In exocytosis, a vesicle migrates to the plasma membrane, binds with it, and releases its contents to the outside of the cell. Exocytosis can be used to remove damaging particles from the inside of the cell or to remove helpful products that have been made within the Rough-ER or the Smooth-ER and packaged in the Golgi. With that, we end our study of Chapter 3. You should take some time to read the book, take notes in your own words, study, and complete your homework.